everybody. You are in the session on wheat digestibility, and we're so happy to see you here today. My name is Harold Stanislawski. I'm the Business Development Director for the Ag Utilization Research Institute. And I want to introduce my dear colleague, Becky Phillip, who is the Program Manager with AURI, and has done an enormous amount of work with this project here. And the purpose of this session is to kind of get you up to speed on what's been going on in wheat research and food science. And so often we hear about folks that have gluten sensitivity. I can't eat bread, I've got gluten issues, or I'm a celiac, and so on and so forth. And it begs the question, what's causing all of the sensitivity issues in a product that Minnesota and North Dakota farmers raise? And is there anything that can be done to reduce gluten sensitivity, not necessarily for celiacs, because that's a very serious issue, but just gluten sensitivity? Is there ways to process the breads better, or is there ways to potentially get different varieties? Becky, we were doing real good until that happened. This project was a partnership between the Minnesota Association of Wheat Growers, and I want to thank the Board of Directors of the Wheat Growers, as well as the Executive Director, Charlie Vogel, for all of the support that they have given in this project. And of course, the University of Minnesota with Dr. Jim Anderson, the wheat breeder, who has been intricately involved. And we also connected with a fellow you're going to meet on screen, Dr. George Annor. He's our food scientist at the University of Minnesota, and has done enormous amount of work with the fermentation and bread issues. And in addition to that, how do these ideas get started in the first place? Usually there's an entrepreneur involved, and we have an entrepreneur in this case. He's sitting right here. Brian LaPlante from Crookston, Minnesota, was really the catalyst who got this project started. And I'm not going to take his thunder away from him, because it's an interesting story about, family, and a person who maybe had some issues, and how Brian tackled the problem by looking at food science as an answer, as almost medicine, if you will, to help alleviate some of the issues. And it's a great story, and I wish his son Ethan was here, because Ethan would be a fantastic spokesman for this. So what we're going to do today is you're going to hear a little bit first about what the University of Minnesota has done on this issue, making wheat varieties potentially more suitable for digestibility, and all the work that went in screening varieties and such. And then when Dr. Anderson finishes, I'll come back and tell you a little bit more about how we intend to move this through the value chain to actually benefit growers, benefit consumers, benefit processors. I'll be back to talk about that. And then Dr. Annor will finish up with the food science part of it and give you a good briefing of what's happened there. So with that, I want to introduce Dr. Jim Anderson. He is a wheat breeder from the University of Minnesota, and he provides guidance to new breeding programs on intermediate wheatgrass, which is Kernza, field pennycress, and camelina, as well as, of course, spring wheat. The research on each species includes a multiple cultivar development and investigation of breeding genetic principles relating to all these crops. So with that, Jim, I'm going to turn it over to you, and let's welcome Jim Anderson.
All right, thank you, Harold. Well, I'll just get right into it. But this is a topic that I've been interested in doing research on for a number of years. So when Brian came to us with this idea, it's like, okay, let's get into this and see if we can make some progress here. So we all know that wheat consumption in the U.S. has been on the decline for decades, actually. So if you remember the Atkins diet, that was one of the big ones. And now it's a lot of folks just going gluten-free, there's different fad diets that are excluding wheat or other grains altogether. But the big thing is just the avoidance of gluten or avoiding wheat in people's diets, whether they have a good reason to or not in some cases. So why are people doing this? Gluten is a protein that's found in wheat, barley, rye, so the close relatives of wheat. Celiac disease, that's an immune disease. It's only about 1% of the US population that truly has celiac. So that is, that's not the majority of why people are going gluten-free. There's also wheat allergies. That's probably a few more people than celiac, but still is going to be a very small percentage of the population. So what's really causing these sensitivity issues when people consume wheat and wheat products? And it's called non-allergy, non-celiac wheat sensitivity, or NCWS. And it causes all sorts of conditions, but just kind of this bloating feeling, or you just don't feel very well, other gastrointestinal issues. So if it's not the gluten itself, what could it be? What constituents in that wheat grain could be causing this? And we focused in on two, and there's some literature to back this up. So we're not the first to go down this road. Others have investigated this already. So FODMAPs is an acronym for fermentable, monosaccharides, and polyols. And ATI are Amylase Trypsin Inhibitors. So it could be one or both of these, and this project intended to investigate both of these, what levels they're at in our current wheats, and is there a way that we can reduce them either by breeding or by processing methodologies. So FODMAP levels in some common foods are shown on this graph. And we've got a lot of wheat products over on the right-hand side here. So this is in grams per serving. Kind of that target value is 0.3 grams per serving or less. And you can see most things with wheat are much higher than that. Rice is very low in FODMAPs. If we go over to the left side, some other foods, garlic is kind of notorious for being high in FODMAPs. Onion is high in FODMAPs. So if you have sensitivity issues to any of those types of foods, then it could be due to FODMAPs. And if our wheat is too high in FODMAPs, maybe that's part of the issue. So these are the folks that put in a proposal to MDA to try to make some headway here and figure out what we can do about this situation. So long-term goals: reduce the discomforts resulting from consumption of wheat-based products, improve consumer health, and increase profitability for wheat growers. So this is going to be good for the wheat industry if we can find some solution here. So 3 objectives. First one is the one that I'm going to be talking about, characterizing the genetic variation in the wheat that we're growing, and then we went back and looked at some heritage wheats as well, seeing if breeding has increased them over time. So is breeding part of the problem or part of the solution, essentially? Second is the processing side, using fermentation. Can we reduce the amount of FODMAPs or ATI in finished products that are going through a fermentation process? And then finally, establish a pathway for industry to implement these research outcomes. So, objective 1, the wheat. So, I quickly assembled a panel of 220 wheat lines, and I'll show you the breakdown of those here in a minute. We grew them at 2 field sites in Minnesota, Crookston and St. Paul 2 years ago. We did genetic marker work on them, basically get a DNA fingerprint for all of them. And then then we get the FODMAP and the ATI values using chromatography techniques. That's in Dr. Annor's lab. And then we take the genetic fingerprints, the FODMAP and ATI values, and do something that we call association mapping. That's where we try to find out what's the genetic cause, or are there specific genes within the wheat that are responsible for those ATI and FODMAP values. So here's the description of the panel of lines that we used. I wanted to make sure we had a lot of what I would call heritage wheats. These are broadly defined as wheats released before 1970. 1970, I chose that because that's the time when the semi-dwarf genes first came into our region. So the variety era was the first semi-dwarf that was popular in this region. So we had quite a few 46 older wheats, 142 modern ones. And then these other ones I just wanted something a little bit more genetically distant, so they're sort of outgroups. So we had 5 Durhams and progenitors of wheat. So wheat is a hexaploid that has 3 genomes, the A, the B, and the D. Einkorn is a diploid, it has only the A genome. Emmer is similar to durum, it's a progenitor of durum, it has the A and the B genome. And a synthetic hexaploid is made by crossing an Emmer with something that's not on the screen that contains the D genome. So it was more of a curiosity to see where these guys would fall. So we grew these out in St. Paul and Crookston, probably the most diverse group of wheats I've ever grown in 20-some years here. Question?
I'm just wondering, with the modern wheats, did you try to get varieties from each decade, or is it kind of stacked heavily towards the last 5 or 10 years?
The modern wheats there's a good representation from every decade, a little bit more focus on the modern ones that represent kind of our current breeding program and what we're crossing with. So getting right into the results. So first thing we did is look at the genetic diversity. So this is basically a scatter plot in 2 directions, the x-axis and y-axis. And the farther apart the points are on this graph, the more distantly related they are. So even I can't read these, but that's not the intent here. But basically all of the bred wheat, or the common wheat lines, are here. The durums, synthetic hexaploids, are here, and the emmers are over here. So kind of what we expected. So the next graph, these guys are going to be gone, and it's just the bred wheat separating out them. And now we can see very good separation of individual varieties. All of those modern lines, 1970 or later, are over here. The founder or heritage wheats are over in this quadrant. And then kind of introductions to our program over the years, mostly from Mexican wheats or from China. That's where we got our scab resistance genes from. It's from Chinese germplasm. They kind of fell up in this quadrant. So very, what we're looking for is a nice broad spread here. It tells us we have a lot of genetic diversity in our panel. So this slide took George Annor's lab months and months and months, and under COVID restrictions, so I don't feel like I'm doing it justice showing all of these data points in just one slide. But here it is. On the x-axis are on the x-axis are the FODMAP levels in percent. So they're going from 0 to 2%. On the y-axis is ATIs ranging from 1 to 4%. The main point here is that we have a lot of variation in this material. And if I were to put kind of a target value on the FODMAPs, 0.3% is about where we would like to be. So among the current varieties, or even I'm not sure which ones these are older or the newer ones, but very few are less than 0.3, so they're more on the higher end. But as a breeder, I see something like this, and if I can see that there's genetic variation there, we can breed either direction. We can breed it lower, we can breed it higher. This graph is showing all of that genetic marker work. So let me take you through it. A lot of pretty colors here. On the x-axis are the 21 chromosomes of wheat, and within each chromosome, there's hundreds, if not 1,000, different dots. Those are individual DNA markers spaced along that length of chromosome. The y-axis indicates the association of every particular marker with that trait. In this case, it's FODMAP. So something that's up high, that means there's a gene on chromosome 1 that is associated with FODMAPs. But this is the significant statistical significance threshold at a 3. So ideally what we would like to see is some that are way up here off the screen at 10 or 12. If we had that, then we could say, oh, there's one or a few genes that control this trait. What this graph is telling me is we've got all this kind of noise in here. There's probably a lot of genes of small effect that are controlling this trait. It doesn't mean that we can't breed for it. It just means that it maybe will take a little bit more time, and we have to use some different approaches. So that's for FODMAPs. For ATI, it's a little bit more promising. Here's that 3 significance threshold. We have a number of things above that. This one is maybe of some interest. That's probably going to explain maybe 10% or so of the variation in ATI. So if that's something we could breed into new wheats, we could maybe lower the ATIs. All right, here's the graph that's of most interest to me coming out of this, all of this work. And the heading kind of says that the FODMAPs and the ATIs have not increased over time. So these are release varieties from our wheat breeding program going back into the early 1900s, all the way up to modern releases. And if we draw a line here, if anything, the FODMAPs are actually going down a little bit. So that's going the good direction. ATIs are maybe going up slightly, but the ATIs are also correlated with the protein. So as the grain protein goes up, the ATI values are going to tend to go up also. And we've released more higher protein varieties in recent years. So I think that's responsible for this uptake here. So this was sort of a huge moment for me, because it's like, okay, the wheat breeding itself, at least in our program, is not to blame for this. We're not seeing, we haven't doubled the amount of FODMAPs in the wheat inadvertently, at least. Okay, so to summarize this portion, and then I'll turn it over to George to talk about the fermentation part of it. We have a genetically diverse set of wheat lines that we're analyzing, so I think it's a proper set. We're seeing a lot of variation in the FODMAPs and the ATIs, about a 3 times variation in the wheat for FODMAPs, 2 times variation for ATI. So as a breeding program, I think that's something we can work with. The einkorns, I didn't present them in the slide, but they were low in both ATI and FODMAP, kind of down at that target level of 0.3. So if somebody's growing ancient wheats, that could be a marketing opportunity for you. Emmers are also low in FODMAP. So that tells me that there's genetic variation in those species as well that we could possibly incorporate into modern wheat. We also found no genomic region, so those graphs with the colors, no region is responsible for a large portion of the variation, but we can still breed for lower values if we choose to do that. And finally, we didn't see any patterns regarding year of release. So the breeding process per se has neither increased nor decreased FODMAPs or ATIs over time. All right.
Becky, can you hear me?
Go ahead.
Okay, George, let me introduce you quick. So George Annor is assistant professor in the Department of Food Science and Nutrition and the General Mills Chair in Cereal Science, I think is his title.
Yep.
All right, go ahead, George.
All right. Thank you, Jim. So I would take over the fermentation part of the second part of this work, which was on fermentation. And the whole idea for this was to give consumers an opportunity to do something about being able to reduce FODMAPs or ATIs in wheat. Now, this part of the work was done in collaboration with Brian of Backward Foods. He actually did most part of the work. I did the analysis part. So what was the objective here? We wanted to explore whether sourdough fermentation can be used as a means to reduce the amount of FODMAPs and ATI. So what did we do? We actually prepared sourdough from these wheat varieties. And these varieties were selected based on the levels of FODMAPs. We had low, high, and medium groups of FODMAPs and ATI, and we wanted to see the extent to which this will be reduced. Next slide. So this is a very nice picture from Rolf. I think it was taken when Brian was doing his experiment. A very beautiful picture showing bread and basically the ingredients that were used for this analysis for the sourdough preparation. It was basically flour and then salt and then the starter culture that is used for making sourdough. So we wanted to keep it very basic and not too complicated. Next slide. So to look at the potential of fermentation to reduce the amounts of FODMAPs and ATI, as I said, we selected about 30 varieties, 10 varieties from low, medium, high FODMAPs, 10 varieties from low, medium, high ATIs. These were grown in both Crookston and St. Paul to also see whether there were any differences in that will be seen due to the environment. And Brian prepared type 1 sourdough, basically using a starter culture from bacteria and yeast. And these samples were fermented for 4 hours and 12 hours. We wanted to see how the period of fermentation would reduce the levels of these FODMAPs and ATIs. And type 1 sourdough was used as a control, so just like times 0, kind of. So that is Brian there, very busy with his experiments. He sent me a lot of samples to analyze. So next slide. So this picture here is showing the ingredients that were used. So it's basically flour from these wheat varieties, and also the starter culture, which is in the middle, and this was made into the dough. I would like to state here that we ended with the dough. We did not bake the bread because of time and because it really needed a lot of work. So the results that I'll be presenting is on the dough. Next slide. So 600 individual samples were sent to me. Yep, a lot to be analyzed. These samples were frozen immediately after the fermentation time was reached. And when they were brought to me, they were all freeze dried. We had to dry them to be able to do the analysis. Because of the COVID restrictions and time constraint, we were not able to analyze all the 600. We actually subsampled from that 600, but we subsampled in a way that the objectives of the study were still met. Next slide. So this here is a result of a graph showing how fermentation reduced the amount of ATIs. ATIs are anti-nutritive factors or proteins that are released by plants as a defense mechanism. So in actual fact, plants do not want us to eat the grains that we harvest from them. So these ATIs cause a lot of immune response in people, especially those who have irritable bowel syndrome. Now, when we did fermentation for 4 hours, we were able to reduce ATIs by as much as 18% reduction. So we went from about 2.1 to about less than 2%. This might seem small, but it's still very significant. Now, when we went to 12 hours of fermentation, we even got much more reduction. Now, we could have gone on with the fermentation, but of course, you also have to bear in mind the sensory characteristics of the sourdough bread. So at least this showed us that we can be able to reduce these ATIs, or consumers can just ferment or prepare sourdough bread, and this should be able to significantly reduce the amount of ATIs if they buy wheat that is already high in levels of ATIs. Next slide. In terms of the FODMAPs, we saw that, so this year are 3 results in one. So the green bars are representing what we call fructans. These are short-chain carbohydrates that are made from fructose, and mainly fructose is the cause of all these issues with FODMAPs. Our bodies are not able to absorb fructose very effectively, and therefore they remain in our small intestines, and pretty much in our large intestines, and they cause a lot of irritation. They are fermented by microorganisms. It causes a lot of bloating. Now, we saw that we were able to significantly reduce fructans by fermentation. We went from just under 0.2% to about just about 0.1%. Percentage-wise, it's about 18 to about 40% reduction. And that is pretty significant in the levels at which we were able to reduce these fructans. Now, if you look at these other graphs, so the blue, gray or blue represent FODMAPs. So you would see that when we did ferment the dough, we saw a significant increase in FODMAP. We saw huge increases. And the first time I saw the results, I was like, oh, what have we done? We thought we were going to have a reduction. But when I looked at the data pretty well, I realized that we also saw an increase in what we call polyols. These are sugar alcohols that were produced during the fermentation of the dough. And it makes sense in a sense that the yeast is actually taking these fructans or fructooligosaccharides and converting them into sugar alcohols. And the good thing about these sugar alcohols is that when we bake bread, we lose all these or almost all these sugar alcohols. So we see this increase because it is in the dough. But of course, we do not eat the dough unless people want to eat frozen dough. That is a different thing. But if we bake bread, we should have a significant reduction in the amount of FODMAPs. So if you should ask me whether fermentation reduces FODMAPs in sourdough bread, I would say a resounding yes, because fermentation would do the job for us. Next slide. So I would hand over to Becky to talk about the establishing the pathway for industry, and I'll come back again with our next steps.
Okay, well, thank you, George. And one of the aspects of this project that was funded, by the way, by the Minnesota Department of Agriculture, and we need to thank them for the funding, was that we have to have some outreach on this program. We have to say, where at the end of the day do we want to go? So I just want to talk to you about the outreach that was done on this project, and then allude a little further along of how we might get it into the marketplace and get it more pronounced in the marketplace. So first off, outreach efforts were led by AURI in partnership with the University Regional Development Partnership, the researchers, the Research and Promotion Council, and background foods. So always on these kinds of projects, we try to keep everyone together that participated in all this research. So the project promotion began in 2019, and it focused on farmers, the Viva the annual Prairie Grains Conference, and then last year, of course, with the COVID, most everything was done through Zoom, and there was a considerable amount of that occurred. And we also reached out to the indigenous populations and the underserved ag producers with the opportunity that might exist down the road. Research overall was broadly targeted to reach farmers because that's what the Minnesota Department of Ag requires, but at the end of the day, you also have to talk to the processors and the consumers and so on and so forth. So that was done again through other avenues of the Food Council, Wheat Food Council, the American Bakers Association, the Institute of Food Technologies, Northern Crop Institute, and a number of webinars that occurred. And it was really interesting because we had participation from Canada. I think, Becky, we even had some participation from another country, if I can remember, and all over the country in this. Because again, this type of project, I mean, gluten sensitivity is something that everybody seems to understand because we almost know, we know a lot of people that have that. Industry and research focused brochures and all of that was developed, and you can see that on the back table back there and pick up a copy. But now I want to talk about a little bit of, we also reviewed a USDA program called Process Verified. And there are all kinds of certification programs. You've heard of organic certification, just a whole host of them out there. But the USDA also has something called Process Verified. I want to talk about that. A little closer to the mic. Should I hold it up?
Yeah,
Okay. Next slide, Becky, please. All right, so what is already in the marketplace on this regard? We're perhaps a little bit behind. Australia, for instance, already has certified food products that are already low FODMAP certified. And that occurred back in that country for quite some time already. They've done the research. Monash University, and Brian can speak to this later, was one of the first ones who got some of this work done. So a lot of products are out there. And they have this low FODMAP certification project in Australia. So therefore, the consumers can be assured that products they're getting are truly low FODMAP through the certification. And now, when we talk about USDA Process Verified, I'm going to show you a pathway of how that can be accomplished here. Next slide. So here's Australia's example of how this is done. They've developed a program called Low FODMAP Certified. It's trademarked. They have been here in the U.S. and they also have products that are sold in the U.S. under this trademark. So as sophisticated consumers, and there are a lot of them out there, and they understand this, they seek this kind of thing out and they purchase it. Next slide. Here's an example. This is the Madrilla Group that operates Australia's largest flour mill. And they are currently selling their low FODMAP flour in the United States. Next slide. So the future is bright. The research results will benefit the value chain, the wheat industry, and the consumers. And we are looking at USDA Process Verified. Here's how this is going to happen. As we get into phase 2, which will be talked about by George here in just a few minutes, we are already seeking out both artisan craft bakers as well as mainstream bakers to look at this USDA process certification. So as it goes through, for instance, fermentation times. And Brian will show you some breads up here that are fake in terms of sourdough. They're not sourdough at all. They're not certified, they're not verified, but a consumer picks it up and says, boy, I'm getting true sourdough bread, and they're not. The only one that is this one. Brian can talk about that. So when we do this, when we get our pilot projects up, we're going to work with a USDA inspector out of Minneapolis that runs the USDA Process Verified. We will have some early entrance into this that will then have a product that truly has the characteristics that the consumer is looking for, which is low FODMAP, low fructan, and all the rest of it. So with that, George, I am going to turn it back to you to talk about where we're going with the next steps. And George is going to make sure that this all happens. Thank you, George.
All right. Thank you, Harold. So we are not ending here. We're very happy with the results we got from the phase 1 of the study, where we realized that we can be able to amend or breed for wheat that has low FODMAPs and ATI, where we also concluded that with fermentation, we can reduce ATIs by as much as 44% in 4 hours, 70% in 12 hours, and reduce fructose by as much as 18% in 4 hours and 40% in 12 hours. So next slide, please. So the next thing we wanted to do was well, we've already proposed and submitted a proposal to the MDA, which hopefully would be funded. But the whole idea is to build up on the phase 1 and move from breeding wheat that has low FODMAPs and ATIs and adding another angle to it, another angle of having high amylose and high resistance starch, which would in effect have high fiber. And I will talk about the reasons why we need fiber and the reasons why we really are interested in doing this work. But the objective for what we proposed is to evaluate the effect of different amylose levels on ergonomic performance and dough functionality. Yes, we want to increase amylose content, but we want to be sure that we don't just increase amylose content for increasing sake. We want to increase the amylose content to levels that would not negatively impact yield and that will not negatively impact performance, because that will obviously affect farmer productivity. We also want to now develop and evaluate wheat germplasm that has high amylose, which will result in high fiber, but also has low FODMAPs and ATI that would increase or improve digestibility. So this will really be good for consumers. Now, we were very lucky to be partnering. We are very lucky to be partnering with WGRC based off in Kansas, and they have a lot of wild wheat germplasm. And if you remember what Jim said, even though we understand the FODMAPs, we still think we have a little more loopholes to close up. And we think that analyzing these wild wheat germplasm would really give us a very good, a better understanding of the genetic link between FODMAPs and ATIs. The good thing about this third objective is that the WGRC is ready to donate or contribute $50,000 to this project if we are funded. And then, of course, we have to do the same thing of reaching out to the food industry, reaching out to farmers, not only farmers, but emerging farmers with the results that we find, and also moving on to protocols for getting the process verified, USDA process verified. Next slide. So why resistant starch, why amylose? So resistant starch is a portion of starches or carbohydrate that escapes digestion. So when you eat any food, digestion of the food starts in your mouth. Your saliva has alpha amylases that immediately start digestion. The food travels to your stomach, it travels to your small intestines where the digestion keeps going on. You have enzyme pancreatic alpha amylases. Resistant starches are starches that escape all these digestion processes and reaches your large intestines without being digested. And when they get into your large intestines, they actually simulate the growth, or they stimulate the growth of good bacteria. And therefore, there's a lot of metabolic activity of these good bacteria. And we know sometimes they can actually suppress the growth of bad bacteria. Resistant starch intake is very low in the US and very low in most part of the world, because we are eating a lot of refined processed foods. Now, if you take our wheat samples that we sell on the market, most of them are refined. That's why there's not a push towards whole grains and whole foods. But we obviously are not meeting our 20 grams per day of dietary fiber intake. Most people meet about 10 grams per day. And therefore, we believe that increasing the resistant starch in wheat, which is consumed a lot by people, would have a major impact in increasing dietary fiber consumption in consumers. Next slide. So we already know that there are some wheat varieties on the market that have high amylose, and experiments have shown that some of these wheat varieties actually do have about 10-fold increase in resistant starch. And this increase in resistant starch obviously results in an increase in dietary fiber, which is very good for us. However, there's not a lot of these varieties. I think there's about just one or two that we know of these high amylose starch, high amylose wheat varieties on the market. But having said that, we think what we are trying to work on is unique in the sense that we would have wheat varieties that have low FODMAPs and low ATI, but also have high amylose. So this has not been done. This is not on the market as far as we are concerned. And we think this would definitely reduce the digestibility issues consumers are having, improve dietary fiber intake, and therefore increase the demand for the consumption of wheat, which will obviously go down to make farmers make more money and help the economy of Minnesota, since wheat is the main crop here in Minnesota. Next slide. Now, I cannot end this presentation without thanking all those who were involved. At the University of Minnesota, we have Emily, Susan, Nate, Prince, Bilola, who helped in doing all the research. During the pandemic, we were working nonstop to get this thing done. And of course, in AURI, Shannon, Becky, Harold, Lolly, everybody else at the station, we have Swan Ray, and of course, Charlie and Melissa from Minnesota Wheat, and Brian from Backward Foods, who did all the fermentation work, without which we probably never would have found out whether fermentation would be able to reduce FODMAPs and ATIs. Next slide. To learn more about this project, I think you can get onto the website of AURI. We've done a lot. They have a very comprehensive or shortened report that you can read about this. You can call Harold. You can also reach us at the university, myself or Jim Anderson, and we'll be very happy to help. We cannot say a big thank you enough to Minnesota Department of Agriculture for funding this whole study in the phase 1. And we hope to get another round of funding for the phase 2, because we believe it's really something we have to tackle when it comes to wheat consumption and wheat demand here in Minnesota. Thank you very much.
Okay, very good. So I think what we'll do is, first off, we've had some other folks enter the room, and Molly and Annie and Paul, if you want to come on up here, it'd be great. We're going to have the sourdough platform up here, and just to participate and ask questions and tell a little bit about what you guys are doing too. So come on up. Molly and Annie and Paul are with Three Farm Daughters right here in Grand Forks, North Dakota. They opened up the door so we could get in today. So glad to be here in your town, Paul. Oh, Grace and Molly. Okay.
It's okay. We all know.
I get confused, but we're gonna turn it over now to the sourdough platform, and what we're gonna do up here is we're gonna talk about these breads. We're gonna talk about the sourdough fermentation process. You're gonna have an opportunity, maybe even to come up here and taste some of these. But first of all, talk about the philosophy of all of this and why it's important in the marketplace. So, Brian, I'm going to turn it over to you to kind of start this thing out, and then we'll have a little discussion with Three Farm Daughters as well, who are in the pasta side of the business. So.
Well, very good, thank you. I think I'm a little plant, so everyone will probably hear me just fine.
If you need it, we'll take it.
Yeah, that's fine. You know, we started this project a number of years ago by kind of answering people's questions as to why in the past were people able to eat bread products and cereal grain products. Why are we struggling so much right now? And so much of the discussion was on gluten. But knowing that gluten really affects a narrow population, a tiny percentage of the population, why were we seeing 25, 30, 35% of the population dropping out of cereal grains? Even upstairs this at lunchtime, it was amazing, overhearing some folks saying, I can't take the buns, I have a gluten issue. And you know that people are having legitimate reactivity to wheat and cereal grains, but why is that happening? And that was the impetus for this research project, is to, number one, find out what's happening in the wheat itself. Because a lot of the discussion was, are we breeding ourselves into reactivity in wheat? So Dr. Anderson addressed that. And Dr. Annor also addressed the issues of what happens in fermentation. So that's kind of where we are today. We know that a large portion of what explains reactivity to people consuming wheat now really comes back down to the processors. The thing that's really changed over the last 100 years isn't so much the wheat. It's not the farmer's fault, it's not the millers, it's not the breeders, it's the processors. And what's happened since especially after World War II, you know, in the early 1900s, sourdough fermentation, authentic type 1 sourdough fermentation was still utilized. But after World War II, you know, things really changed. Changed on the farm, changed on processing, faster food production, more food production. That was the goal, but we didn't really understand what we're leaving behind when we're doing that, right? We're speeding up food processes, and we didn't really understand what was going to happen. So food processes back in the 60s and 70s that might have been a 3 or 4 hour yeast only type of process would address some of the digestive issues. But now today it's rapidly speeding up. I mean, it's not unheard of for commercial processes of breads to be an hour process. It's largely a whipping process, not a lot of fermentation going on. So what explains most, I believe, of what's happening with consumers really ends up being how it's processed in the finished goods. So, of course, in the last 7, 8, 9 years, there's been a lot of interest in sourdough. A lot of that comes from Europe, a lot of it comes from Dr. Gänzle's work, and a lot from Australia. And so people are interested in sourdough. And a lot of people come up to me and say, I really love sourdough. I get it, you know, it tastes great, I feel better with it. But what you'll see is, Harold picked up some bread for us. That loaf right there is just a standard modern bread, doesn't claim to be sourdough. This loaf right here is a sourdough sandwich bread from Walmart. And so you say, great, I'm buying that. Here's another one made from an artisan bakery that claims to be sourdough. What we have to start looking at now is what the label says. So an authentic sourdough is usually, if it's a type 1 culture, which is what I use, is bacteria and yeast maintained in a flour and water culture, and it's refreshed and it's used as the leavening, as the fermentation of the bread exclusively. No additional yeast, no additional enzymes, nothing. So a French loaf of bread like we make is just flour, water, salt. And the culture is just water and flour. So the bacteria, the yeast that exist, that are refreshed and repopulated and kept at a high population for leavening and fermentation, that's what sourdough is. If you have to look at a list on an ingredient list and it says yeast, or it'll say ascorbic acid, or fumaric acid, or lactic acid, they're adding acids to create a flavor to simulate sourdough, but it's not sourdough. You're not going to see the degradation of the anti-nutrients. You'll get some of the benefits of lower pH, which has its own benefits. But it's not.
Right, I want to pass those bolts around.
Sure, go ahead.
Just so you can see what Brian's talking about, because you have the nutrition facts in the back, and if you look on the ingredients, you can tell immediately that this is not real sourdough bread, but it says sourdough.
Yep, and so we've discussed that today. One of the things that's important moving forward, so that consumers know they're going to pick up something that's actually going to digest well, and give them the amazing health benefits from fermentation, is kind of a truth in labeling. So we want to really pursue process verified from the USDA. And that's pretty simple and straightforward, because all we really need to say is, did you use a live culture? Did you ferment this using the culture? Yes or no? And that's what process verified can do for us. So the consumer can pick up something just like you do certified Angus or anything else. You can look at it and say, I know by this USDA label that this is authentic sourdough. So that the other ones you're seeing around here can't do that. It's important. So, you know, I just brought some quick little samples here of what a culture looks like. You can come up and see it when you get a chance. A lot of what happens in here, you know, George was talking about ease of digestibility. A lot of pre-digestion, if you want to call it that, happens during your fermentation processes. So you'll see a lot of bubbles in here. That if you eat a loaf of bread that's not fermented, a lot of what's going on here is going on in your system, and air, that burst of air, if you want to call it that, the gases that are released causes a lot of problems in the digestive system. And what we know now is that based on the bacteria and yeast that are in your own digestive system, will determine a lot of how reactive products and cereal grains are to you. Some people can handle it, some people have a big reaction to it, and it's largely due to the population of the bacteria and yeast in your own system. So that's what fermentation is, that's what sourdough really is, and that's why it's important. That's what we wanted to accomplish with this project. So in the future, as George and Jim have said, we want to look at lines of wheat that are naturally low in ATI and FODMAPs to begin with, especially when it's getting into the commercial guys, so that there's less reactivity for consumers to begin with. But then we want to promote sourdough fermentation, so that you can bring that even farther down. So we want to kind of recreate what was done for literally thousands of years that made things very digestible for consumers. So sourdough has a lot of benefits. You're gonna increase the availability of amino acids. You're lowering the pH of the product so that it's gonna have a better reaction for blood sugar results, insulin results, glycemic response, glycemic index. Those are all important things. Mineral availability is greatly enhanced. So many things happen within sourdough fermentation, and Dr. Gänzle can talk forever on this because he's really the leader in the world, in my opinion, in fermentation research and the microbiome. There's so many things that happen within fermentation that as you look at food in the future, it's going to be geared toward what does that food do to the bacteria, the good bacteria, the good fungal entities in your system. That's what keeps you healthy. The goal is going to be to feed the bacteria to keep you healthy. And that's a lot of what sourdough and fermented products do for us. So in a nutshell, that's what sourdough does and why it's so important. We will, as a company, be developing a number of products that will be released on the market commercially. So we've developed, of course, breads. We're developing pizza crusts that will be for the frozen market. Pasta is really important, huge market there. And you know, things like English muffins, desserts, all those things that used to be done with this technology forever will be brought back onto the market. So I'd like these folks here who are doing some important work on resistant starch too, which is what George and Jim are talking about. Their work in the high fiber, high resistant starch is the other element that's so important in cereal grains, and I'd like them to speak some about what they're doing, what they're pioneering. It's really important.
Thank you. So I'm Molly, this is my sister Grace, and our father Paul. Unfortunately, our third sister couldn't be here today. But we're the three farm daughters. We're based out of Grand Forks here. Our parents are actually farmers in the Red River Valley. And what we have come to do is we have created what we have done is taken a high fiber wheat and created our pasta line. So we sell high fiber pastas in retail and then online. And just what we've got to really know and learn in the short year that we've been doing this is that the customer response is immense. And the need for, not only healthier products, but products that are filling a niche that people maybe didn't have prior has been amazing. And so kind of learning what AURI is doing and the whole group here with growing and just what they're all doing is really interesting to us, because as you can take this high pasta and the resistant starch wheats and just even make it kind of a new product. And so, the work that they're doing to even look at the process verified would be really interesting to us as a kind of the retail side, because as we work on it, you see a lot of labels like USDA organic or non-GMO project or all these different labels, and then putting like a real stamp on it of approval through the USDA or whatnot is important, because there's a lot of claims and companies that can make claims, but doing that verified process is very, very interesting and very direct and transparent with the consumer, in my mind.
And one thing to add too is that the market, people are extremely educated on things that make their own bodies feel good and what doesn't feel good. And like Ryan saying just at lunch today, noticing people that are avoiding grains, and it's just such a small population that actually have the total intolerance, that actually have celiac. But so many, just because of the way that grains are currently being produced, are having that gut distension and like the really bad bloating and pain. So what Brian's doing is really an amazing project because it's people that may have just a sensitivity but aren't necessarily allergic who get to enjoy the foods they love getting. So it's really, really amazing.
And one of the things this pasta does when it's made, with the grains that we're using, the blends that we're doing, you're running 8 to 9 grams of fiber per serving, where a normal serving of pasta is 2. So there's a big difference there. It's a help for a threshold to get your daily fiber. I read one time where women need 25 grams and men need about 30 to 35 grams of fiber in their diet. And the other thing about this pasta, when you do cook it, it takes a little bit longer, a few more minutes. But when you put it in your fridge and leave it there, you take it out the next day or a couple days later, it has that look and feel like it's just been cooked again when you microwave it.
To kind of summarize it, it's a spectrum here, and it's interesting because what we've attempted to do on this research project and what Three Farm Daughters are doing is really important because the two huge benefits of wheat, the nutrition and what can happen on the fiber side as a complete food, is really important because you'll hear over and over and over again with the Mediterranean diet, with the Blue Zone diet, all these things, fiber and whole grain nutrition is critical to human diets. And a lot of people have cut that out of their diet. And there's unique characteristics about wheat that can't really be easily replaced. The selenium content, all these types of B vitamins, a lot of nutrition that occurs with wheat has to come back into human diets. And so I think you answer reactivity, you answer fiber. You get both of those, you lower reactivity, increase fiber, and you bring a whole segment of consumers back to the cereal grains market. It's critical. And it's critical for their diet. And you're going to hear a lot of discussions in the next 5 years and now even about microbiome. You're going to hear microbiome. And it's all about feeding your gut, feeding the live bacteria and yeast that exist there for optimum health. And whole grains for 30,000 years have been a staple of human diet because of that. And we kind of reversed course here in the last 15 years, but we have to come back to it. It's really important.
Brian, I've got a question with the sourdough fermentation and what for the Three Farm Daughter folks do, is because sourdough takes a little longer in the fermentation side and all the rest of it on commercial bakeries and so forth, first of all, are they willing to do it even when there's a demand? And then secondly, for the Three Farm Daughters, I know you tested some of this sourdough. Was it like an aha moment or so-so, or tell us a little bit about that?
So if I was to talk about the model of longer fermentation, sourdough fermentation in the commercial market, not impossible, but it will take some changing gears. The artisan market, the small bakeries, I think it's a no-brainer for them. However, commercial markets can adapt to this, and I'm sure Dr. Gänzle can speak to this. Everyone gets a little scared when they hear, well, 4, 6, 8 hour, 10 hour, 12 hour ferment times. Well, that's for the first process, right? But you can stagger it if you wanted to every 2 minutes, 3 minutes, 4 minutes. You can keep a continual process of longer ferment. It's just your first cycle is going to be longer, but these cultures can be refreshed, and they're every 2 hours ready to reproduce. But you can stagger your processes. You could be in continuous production and never know the difference other than your very first cycle. But the industry can adapt to it. It does take some changes, but I think you're going to see it just like we did with the beer industry, where you had Coors and a few other guys dominating, doing one type of beer, and the craft beer market came in, took a big market share. Commercial guys come back and say, we want part of that back. I think it's just a cycle of life. But I think even the large commercial operators are paying attention to this research. And I'll turn it over to Three Farm Daughters, but I'm impressed with what they're doing and what they're attempting to do.
So as far as, the taste of the sourdough and whatnot, we've been lucky enough to try quite a few things Brian has made in the past, as far as whether it be a sourdough cookie or you've run breads. You've made sourdough pastas. And truly, I truly don't feel like there's a distinct taste that, sometimes I feel like you hear sourdough and everyone's like, oh, it's sour. Or it has this off taste. And when you kind of make the pastas that we've tried with Brian, I don't feel that way. And like the cookie, it was a chocolate cookie and it was delicious. So I think sometimes it's almost getting off that almost like a stigma, but yet people associate sourdough with artisan bread a lot. But I think to Brian's point where he was just saying it can be a commercially viable process, it would just be getting the processors to adapt to it and go that way. So it'll be interesting to see if the cycle come through.
Are there any questions from the audience for Brian or the Three Farm Daughter folks about this whole process? I know, Brian, you're going into the pizza crust business, and I think that's going to be a big demand over there. What I see in the marketplace, when I was just in Alexandria the other day, they were sold out of the fake sourdough. Now, well, people didn't know that it was, but it was just sold out. The rest of the bread was still there, but that part was sold out. So I think that in and alone is a signal to me that there's a demand for this, and there certainly would be a demand for the true sourdough type one that Brian is talking about. So, any questions from you? I mean, first of all, is anybody gluten sensitive in the group? You are. How come?
About 2 years ago, I started having weird breakouts in my hands, and then I started having stomach issues. And I have a cousin who's celiac, so through talking with him, did some experimentation and found out when I cut out gluten, I stopped having outbreaks. It's bad. I got one going on right now. I'll show you later.
Well, Doug, we're going to make sure you get a slice of Brian's sourdough bread and see if there's anything happening.
Yeah, it's weird. Yeah.
How do the heritage grains respond to the sourdough process versus modern grains?
Yeah, that's a good question. Actually, it's indistinguishable. It's really pretty indistinguishable. They function almost ideally. Really, all you're really going to see in ancient versus heritage versus modern is the makeup, the gluten itself, the strength of the dough itself. You'll find that there might be subtle flavor differences, and that'll be largely due to the mineral content, that type of thing, but they do beautifully. So heritage lines, ancient lines, Dr. Gänzle, I know, has done work in spelt, making low FODMAP breads. Rye can be added, barley can be added. These things are all historically very traditional types of breads. So there's no reason that we can't be looking at any of these types of meats, even the specialty lines, to be utilized in sourdough.
So I actually make sourdough bread at home
Yep.
pretty regularly. For me, it takes all day to go from flours and fermentation to actually having a loaf of bread, from like 8 in the morning to 10 at night before
Yep.
I have a loaf of bread. So in a commercial process, is that timing similar, or can you
Well,
adjust things to make that timing faster?
I think there's optimization, and the reason actually we looked at 4 and 12 hours is that generally, if you talk to your great grandma or you knew those folks going back and back, the old rules of thumb of when it doubles, it's ready for this. When it doubles again, it's ready for this. Those sequences for bread loaf optimization volumes, I wanted to target those things to find out what's happening with ATI and FODMAP, because you want a nice product, right? You want something. So yes, there's going to be certain times that have to be met in order to accomplish degradation and also quality. But let's say that you got up at 7 in the morning and you started, you had your culture refreshed and you were going through a whole cycle, but you said, you know what, instead of making 4 loaves of bread today, I want to make 12. You could, right? Because your culture would be divided and divided and divided and maybe refreshed so that you can have everything ready for cycle after cycle after cycle. That's what would happen commercially. And you can make a continuous cycle out of it if you really want to.
Thank you. I'm curious about the pasta. So is it still a durum pasta, or are you doing blends with different wheats?
Yeah, I did a whole range, and I got some product from Sprouls on what they were working on in the high fiber durums, and it's a spectacular product. It makes a wonderful noodle. It has a great flavor to it, And I don't really think you can do better than that. I've done blends of spring wheat, AP flours, bread flours. You can do just about anything, but I have to say that what they're creating over there makes a superior noodle, especially when it's fermented. A great flavor, great outcome. So yeah, you can do a lot of things, but there are some things that will shine.
So we've done it with, to his point, we've done it with the durum. There's a high resistance starch durum, and then we've also done it with a wheat. So blends with a durum or wheat, kind of just figuring out what your optimal recipe would be, really.
I'm curious, I haven't heard of the three farmers' daughters before. What kind of scale are you at in terms of both wheat you're using and how much product you're selling, and then are you milling it, or are you buying?
So that's a great question. We started about a year ago is when we first hit the shelves. So today we're sold in Grand Forks, Fargo, we're in Kowalski's in Minneapolis. We're in about just over 30 some retail stores, and then we sell online and on Amazon. So what we've done is we've worked with, we've actually worked with the North Dakota State Mill to mill some of our stuff, and then just other manufacturers to kind of produce our products. We're really excited. We're just launching, coming in the next couple months, a kind of a rebrand and a refresh. When we started this project, we started with like clip art in the boardroom, making our logo and whatnot. And to Ryan's point, it's like it has really taken off, and people really do search for different nutrition in pastas and breads and whatnot. And so we've got a great response, and that's what's really prompted us to do kind of a whole brand refresh that we're really excited to relaunch in the next couple months. So
People love pasta as is, and you really saw the market shift to alternatives, be that rice pastas or chickpea pastas. But at the end of the day, people still love traditional pasta as it should be. And so we've been excited to be able to bring that to people, kind of similar to what Brian's done, to get to enjoy it with better taste, texture, the same performance that they know and love. They don't have to learn a new way like they would maybe with chickpeas or anything else. So it's really exciting, and we're happy to be here, honestly.
Brian, or any of you, if you're going to make a sourdough pasta versus a regular pasta, what is the difference in the potential cost to the consumer to get one versus another?
From my aspect, as far as a sourdough process, again, it's going to be the time of the process, but there's not a huge difference other than the initial run, the very first run. Because after you start sequencing for continuous production, the only thing that's different in the pasta industry now versus what would have been 100 years ago versus what I would do is that pasta today is made as a dry flowable flour with water under vacuum extruded, right? There's no fermentation, there's no process. It's just done immediately. Our process will have, again, fermentation time and then extrusion. So there will be some adaptation to equipment, but other than the ferment time, which again you can stage for continual production, I don't foresee a tremendous cost difference, just a change in how the equipment is utilized.
Well, that's good news, in my opinion. Just one further question from me, unless nobody else has, but it's our understanding that in Europe, they use a lot more sourdough than we do, for sure. And I routinely get folks who come back from Europe and say, I could eat that bread, it wasn't sick, I didn't get bloated, all the rest of it. And they come back here and they say, no, I'm not eating any bread. So with wheat acreage, declining wheat, to put that back up, it seems like this is a logical way to do that by answering what the consumer seems to be asking us, is to produce bread that they can digest better. Would you all agree?
Yeah, I hear the same thing because I have family that for business travels to France, Italy, Germany, and you hear the same thing. And they're so frustrated when they come back home because they can go just about to any bakery there, get a loaf of bread, and they say, wow, I love it, tastes great, it doesn't bother me. And again, that's what consumers are looking for here, but we have to make it authentic and legitimate, and it has to be recognizable that's why process verified is so important. We cannot blur the spectrum here and confuse consumers. That's really important.
Well, all right, we're right on time and on schedule, Becky. Let's give these folks a hand, and please ask them questions later on. I want to introduce Dr. Michael Gonzale. He's going to be giving us a presentation on food member fermentations for improved digestibility of plant foods. And he is a professor in Canada Research Chair in Food Microbiology and Probiotics, a distinguished professor in the Department of Agriculture, Food and Nutritional Science at the University of Alberta in Edmonton, Canada. And we're really pleased to have our Canadian friends join us today, even if it has to be by Zoom, Dr. Gänzle. So welcome.
Thank you for the introduction. In today's presentation, I want to highlight some of the results that we got on removal of FODMAPs, Amylase Trypsin Inhibitor, and the wheat Germ Agglutinin in wheat fermentations. But also I want to put it in a little bit of bigger context on why is fermentation important for any society that relies on crop agriculture. If we go back in the history of using sourdough, sourdough use starts probably before agriculture started. And for the better part of human history, sourdough was the only leavening, which means anytime a culture produced leavened baked goods, it was a mixture of lactic acid bacteria and yeast that was doing the gas production, what we refer to as sourdough. This changed with the industrialization. Also, bread production became industrialized, which means the scale of operation became bigger. And with the availability of baker's yeast, sourdough was no longer used for leavening, but the main aim was acidification of dough, and particularly acidification of rye dough, because rye flours need acidification for optimal bread quality. This has changed again probably 15 years ago in Europe and no more than 5 years ago in North America. That acidification is not an issue in wheat baking, but we see a strong increase in the use of sourdough, both at the small level, artisanal baking and household bakers, but also at the large industrial baking. And the main aim is now improved bread quality, quality in the broadest sense, nutritional quality, shelf life, and flavor. If we change why we ferment sourdough, we also change how we ferment sourdough and which organisms are in the sourdoughs, and I will get back to that a little bit later. Maybe one picture as to my confidence that sourdough is a fairly old fermentation process. This is a picture of an archaeological site in contemporary Jordan, the Natufian period, which coincides with the time when one of the times when humans started to cultivate wheat, barley, and barley. And at this site also a bread was found or excavated, which was showing clear signs of leavening. I asked the author whether we can have a sample to look for DNA to identify fermentation organisms. Unfortunately, the bread was completely charred and it was no longer possible to get any DNA from the sample. Again, a brief look into history. So sourdough baking and the use of sourdough is approximately 14,000 years old and likely predates crop agriculture. And it remained the only leavening agent until Louis Pasteur invented food microbiology and yeast in 1857. Is there any evidence that bakers predated farmers, or that the ability to ferment was a prerequisite for getting a main part of the daily calories from agricultural crops instead of hunting and gathering? There is some indication that the demand for bread and or beer likely was a driving factor for the domestication of wheat. And then if you think about grain, seeds, and tubers, these are not meant to be eaten by animals, including humans, but they're meant to sustain growth of a new plant. So they're certainly packed with nutrients, but they're also packed with anti-nutritive factors that are aimed to deter insects and animals from eating the seeds. That means without proper processing, they are not really the best solution for human nutrition. If you look at the big picture, going beyond wheat, how does fermentation with lactic acid bacteria change the content of anti-nutritive factors? If you look at this slide at the left, I have the oligosaccharides. That's fructans and fructooligosaccharides, which are the main FODMAPs in wheat and rye. You have the raffinose family oligosaccharides, which are the main family of oligosaccharides in pulses, including soybeans, peas, and beans. And then you have lactose or beta-galacto-oligosaccharides, which obviously is important in dairy products. For all of these oligosaccharides, we have intracellular enzymes, in some cases extracellular enzymes, which degrade the oligosaccharides and convert them to lactate, acetate, and carbon dioxide. So for fermented dairy products, it's well established that it goes a long way in addressing lactose intolerance. It's known that fermentation of pulses reduces any adverse conditions that relate to oligosaccharides. And we are just about to understand what fermentation of wheat does to the fructans. In some plant crops, you have compounds which are outright toxic. The most infamous one are the cyanogenic glucosides, which are present in cassava, which is a main crop in many tropical climates native to South America, but also used in Africa. If these are not removed by fermentation or other processes, cyanide is released, which actually can kill the consumer. So fermentation processes are used to address this issue. Then we have in many of the cereal crops and in the pulses, we have polyphenolic compounds. They have gained a reputation as beneficial antioxidants, but they also taste very bitter and in some cases inhibit digestive enzymes. And lactic acid bacteria possess numerous enzymes that don't completely remove them, but modify them to a point that the bitter taste is less pronounced and likely the impact of digestion is also reduced. If you talk to people in South Africa where they grow sorghum, the sorghum varieties which are highest, which do best in dry climate, the red and black sorghums, they cannot be eaten without proper fermentation because they're simply too bitter. If you look at proteins, there are two mechanisms by which lactic fermentation impacts protein. One is the obvious one, the expression of proteases, which hydrolyzes proteins. The second is a little bit less obvious. Many lactics produce reducing power, which means reduced thiols, which can open up disulfide bonds that stabilize the structure of proteins and thus change their biological activity or make them more susceptible for proteolytic degradation. What does it all mean for wheat sourdough? I won't speak too much about phytate. It has been fairly well established in more than 30 years of research that sourdough reduces the phytate content and improves mineral availability. I will speak briefly about FODMAPs, ATI, and the wheat germ agglutinin. For FODMAPs, the strategy was fairly straightforward. We had in our collection an exceptional strain of Lactobacillus crispatus, which expresses an extracellular fructanase, which degrades oligosaccharides and polysaccharides. So we fermented sourdough with that strain and determined the content of FODMAPs in the resulting bread. This is a very exceptional enzyme which you do not expect in any conventional sourdough. If you look at the databases, we have more than 4,000 genome sequences of lactobacilli in the NCBI database, but very few of those, the last time we checked 6, actually include this extracellular enzyme. So this is a rather exceptional strain. where am I here? So what happens to the fructan content? The bread that we baked was half rye, half wheat. This is the fructan content of the raw material. I indicated it in millimole per kilogram, which is easy if you're looking at biochemistry and metabolic conversions. I will translate to gram per kilogram later. If you compare the number, this strain of Lactobacillus crispatus, which has the extracellular fructanase, achieves a very extensive reduction of the fructans, not only in the sourdough, but also in the bread. Somewhat to our surprise, Lactobacillus reuteri, an organism which is quite frequently found in conventional sourdoughs, but does not have an extracellular fructanase, also achieved a quite substantial reduction of the FODMAPs. Again, this is data in bread, not in sourdough. So this is a reduction by approximately 2/3. We have a little bit addition of increase of the mannitol, but this increase is very small compared to the reduction of fructans. Straight dough bread, and we operate with a basic 2% addition of yeast and a 90-minute proofing time. You still get somewhat of a reduction, but not very substantially. However, if you add the enzyme from the Lactobacillus crispatus to a straight dough bread, you can also achieve a complete reduction of, almost complete reduction of fructans. How does that translate to the tolerance of the product? We didn't do any clinical trials, but if you take the cutoffs for FODMAP, low FODMAP diets that are published out of Monash University, they say a product should have, or a serving should have less than 0.3 grams FODMAPs per serving. If you take the straight dough bread, someone who is sensitive can eat half a slice. With a conventional sourdough, things improve and someone can eat 2 slices. So that's an entire sandwich. With a through A expressing sourdough, there are almost no limits as to how much bread can be eaten. So 4 slices or 2 sandwiches work just fine. What about the amylase trypsin inhibitor? We did 2 studies, one doing a qualitative assessment looking at the ratio of tetramers to monomers. Again, we looked at sourdough and bread. For the sourdough, the results were quite promising because we actually saw a quite substantial reduction of the ratio of the tetramer to the monomer. Justification being that the tetramer is more biologically active than the monomer. However, if we went to the baking, the differences were no longer really big and possibly no longer significant. A little bit later, we developed a mass spec assay to actually achieve a reasonably accurate quantification with LC-MS-MS. That work just concluded this summer. We fermented with 3 different strains, Fructolactobacillus amphotiscensis, a mutant of the same strain, and the homofermentative Lactobacillus cereei. And we always use chemically acidified dough as a control and then straight leavened dough. In all cases, we use sourdough in conjunction with baker's yeast as a leavening agent. We did this with 2 grain varieties. In one, we saw a small difference after proofing. In the other one, we did not. The most important piece is the black bars here. That's the relative abundance in the bread. And you see, if you go from the dough after proofing to bread, you have a strong decrease of the ATI content because the protein denatures, aggregates, and possibly cross-links with other proteins. And any differences between lactic fermented breads and straight dough breads that might have been observed in the sourdoughs was no longer discernible. The last protein that we looked at was wheat germ agglutinin. And here we also looked initially at the sourdough, and that was quite promising. This is the whole wheat flour. This was quantified with an ELISA assay because these are commercially available for the wheat germ agglutinin. This is the whole wheat flour, which contains approximately 6 milligram per kilogram. If you ferment this flour with defined strains of lactic acid bacteria, again, depending on which strain you use, the content is reduced quite substantially, or at least significantly, which doesn't happen in the chemically acidified dough. One thing to note is the difference between these two strains. These are both strains of Fructilactobacillus sanfranciscensis, previously termed Lactobacillus sanfranciscensis, which was isolated first in San Francisco. The two strains are absolutely identical and differ only in one gene. This strain reduces glutathione, which means you get thiol accumulation in dough, and this one does not. And this difference tells us that the thiol exchange reactions in sourdough play a role in the protein structure. Again, I have to pour a little bit water into the wine. Obviously, we also try to extract the wheat germ agglutinin from bread, but after baking, the protein was unextractable and we could not get any useful data. So that gets us to the interim summary. Sourdough baking partially or completely degrades phytate and fructans, so you can make a bread with a strongly reduced FODMAP content. We could show that sourdough fermentation modifies immune-reactive proteins, including ATI and wheat germ agglutinin, but we don't have a lot of confidence whether their content in the bread or the biological activity in the bread is modified. A last piece, if you look at the gluten content, which we did not in this project, but which we have extensively done in previous projects over the last years, the gluten content of sourdough bread is not different from the gluten content of straight dough bread. And I will get back to that a little bit later. Maybe try to link this back to the history of sourdough and how fermentation process are used in baking. We ended with Louis Pasteur, who discovered yeast and its role in food fermentations. No more than 10 years later, the first baker yeast factory opened in Vienna. Probably the real paradigm shift happened only 50 or 60 years later, because since the 1930s and 1940s, baker's yeast was not grown on cereal substrates, where it always grows in conjunction with lactic acid bacteria, but it was grown on molasses, and cultivating the yeast on molasses allowed the companies to produce a fairly pure baker's yeast preparation without too much contamination with lactic acid bacteria. By the time we hit the late 20th century, we had almost exclusive use of baker's yeast for wheat bread and a continuing use of sourdough in rye applications to achieve acidification. I started my work in sourdough in the mid-1990s in Germany. If I was going to a conference in Germany at the time, as soon as I said sourdough, everyone understood rye, and the link of wheat baking and sourdough was foreign to almost all of the audience. If you sum it up, in the 1900s or in the 1800s up to roundabouts 1900s, maybe a little bit later, your ingredient list on bread would be flour, water, salt. That's tradition. This has changed in the 20th century. Baking shifted to industrial baking, baker's yeast as a sole leavening agent, and the increase of scale also required longer shelf lives, and the ingredient list started to be a little bit longer, and we called that innovation. How does that relate to wheat-related immunological or nutritional disorders? Let's start first with celiac disease, and celiac disease affects no more than 1% of the population. This is an auto-inflammatory disease in reaction to gluten proteins, leading to chronic inflammation of the small intestine, and that means nutrients can no longer be absorbed. It's induced in sensitized persons by prolamins of triticum cereals, that is wheat, rye, and barley. All three grains are implicated, and it doesn't matter whether it's an Asian grain or a modern bread wheat. And the limit that is tolerated by most celiacs is 20 milligram of gluten per day. If you look at how much you have in the single serving of wheat bread, that's 1.5 gram. There is very little, if anything, that you can do to safely get from 1.5 gram to less than 20 milligram with fermentation. The story is a little bit different with non-celiac wheat intolerance. The numbers are a little bit unclear. Depending on whom you ask, you have between 6 to 15% of the population. And there seems to be a strong indication that the prevalence is a lot higher in developed countries than in developing countries. It's a poorly defined intestinal symptoms in response to wheat and or FODMAPs. So here it's not gluten that's the problem. It's not necessarily rye or barley that are the problem. So this seems to be more specific to wheat. And we do know that, sorry, FODMAPs do play a role in the wheat intolerance. For amylase trypsin inhibitor and the wheat germ agglutinin, we don't know. There are publications which suggest a role. There are some in vitro experiments or cell cultures or animal experiments which try to establish the link, but all of that was done with wheat flour, wheat dough, or purified ATI. So we really don't know whether ATI and wheat germ agglutinin in bread has a role to play. It's possible or even likely, but we don't know. An important piece is that non-celiac wheat intolerance strongly overlaps with the irritable bowel syndrome and fructose malabsorption. Then comes the big mystery number. If you look at gluten avoidance in the US in these days, the percent of consumers that aim to purchase gluten-free is up to 30%. Those individuals with an obvious and recognized intolerance to wheat or gluten probably account for no more than 15%, which means there is a fairly large segment of the consumers that have no adverse reactions to wheat, but still avoid wheat or gluten because of some perceived negative link of wheat or gluten and health. What can we do with sourdough? For celiac disease, not much. Sourdough does not improve tolerance, period. And whatever we communicate about the benefits of fermentation, we must not bring that in connection with celiac disease because we can help celiacs by using sourdough to make gluten-free bread at a higher quality. We can't help celiacs by fermenting wheat with almost anything, and the few exceptions no more than confirm the rule. If you look at non-celiac wheat sensitivity, the chances are a little bit better. And over the last years, and I spent many phone calls and discussions on this, trying to get my head around it, there is fairly consistent anecdotal reports that imply that Asian grains, rye, and sourdough bread are often tolerated. But until very recently, we didn't have any good signs which would indicate why. We now know, and the publications are no more than 2 or 3 years old, that conventional sourdough fermentations reduce the content of FODMAP. So here we have a true mechanism on why wheat is better tolerated, and that links to the large-scale clinical trials in Australia that demonstrate that FODMAPs actually have adverse effects in some individuals. We know that sourdough fermentation modifies immune active proteins in wheat, but we don't know their content in bread, and we don't know how that links to the role of sourdough fermentation to non-celiac wheat sensitivity. And the big elephant in the room, what we don't know, we lack appropriately powered randomized clinical trials to demonstrate improved tolerance of sourdough. A Finnish company that is producing low FODMAP rye bread, they did 2 small-scale trials, one with mixed results, one with fairly good results, but these may be probably too small to be trustworthy. Getting back to history on what is currently happening, we were stuck in the late 20th century with almost exclusive use of baker's yeast, and probably the increased awareness of gluten and the avoidance of wheat coincides or follows the shift from sourdough baking to wheat baking, but correlations don't tell you anything about causality. What I do know is that since 5 years and probably 10 to 15 years in Europe, sourdough applications in wheat baking are becoming mainstream. So you not only have the small bakers and the farmers market people talking about sourdough, you have the big players that account for a major share of the US bread market that get back into sourdough fermentation. A main reason is flavor and texture. A second main reason is clean label, but they are aware that they also may cut costs and improve the nutritional value. In the large industrial applications, sourdough will always be used in conjunction with baker's yeast, but that still means you have a good share of the flour which undergoes long-term fermentation. So, is the length of the ingredient list inversely proportional to the knowledge of the baker? We started out with flour, water, salt, went to a little bit more lengthy ingredient lists, and I have a lecture in our serial chemistry class. It takes me 3 hours to explain what the ingredients are and why they work. To some extent, we may go back to flour, water, salt. Edmonton was always a place where you had a few small good bakeries that used sourdough as leavening agent and made good bread. But in 2021, for the first time since I came here, I can go to a supermarket and buy bread with the ingredient label flour, water, salt. I think much of what's going to happen in the future will be somewhere in the middle between these two, but we will see an increased use of sourdough in mainstream industrial baking. That's the summary of the presentation, hoping to leave enough time for discussion. Sourdough baking modifies or degrades anti-nutritive compounds, including phytate, FODMAPs, ATI, and Germ Agglutinin. Improved tolerance of sourdough bread in non-celiac wheat sensitivity is reported anecdotally, but not documented in randomized clinical trials. The one area where I have high confidence is the FODMAP degradation. That's one area where I have confidence that sourdough fermentations actually improves tolerance by many people. So I think until we know better, sourdough fermentation remain the best tool to improve tolerance of wheat. We need to keep in mind, however, that not all fermentations are created equals. Different sourdoughs are fermented for a different purpose with different organisms. And this week on Monday, a publication came out that used yeast fermentation to degrade FODMAP. They differed from what's currently done by using a much lower dose of yeast. And to get proper leavening, they needed a much longer fermentation time, and increasing the fermentation time in a yeasted dough apparently went a long way to decrease the content of FODMAPs as well. So last piece, the shift to sourdough baking currently occurs at a big scale. Depending on how fast that is, we may know in 5 to 10 years what that does to non-celiac wheat intolerance and the avoidance of wheat by customers. With that, I want to thank my team in the Food Microlab in 250 Agriculture Forestry Center. We're currently approximately 18 to 20 people, mostly grad students, some visiting students and postdocs. And I want to thank you, of course, for your attention. So thank you.
Very interesting information there. Are there any questions from the floor to the distinguished professor?
Is there a difference in shelf life? I don't know if this is more Brian, your type of question, or one for the professor, but the difference in the shelf life between the sourdough fermentation and the commercial process that's currently used for breads?
I can answer that question because that's one of the topics we are currently working on. If you compare sourdough bread with a yeast-leavened bread with the same ingredient list, then sourdough does better. You have a lower pH, you have a little bit of acetic acid, and some lactobacilli produce specific antifungal compounds. If you compare sourdough bread with a commercial bread which has propionic acid to prevent molds and a bunch of emulsifiers to prevent staling, then the sourdough bread doesn't fare as well as your sliced pan bread. So the industry is a good loaf of sourdough bread. You can keep it for a week and then it gets either moldy or stale or dry. The industry standard for the shelf life of the pan bread is 3 weeks, and with sourdough fermentation only, it's very difficult to get there.
To me, that would be one of the limiting factors to get the market to actually shift over to a sourdough fermentation process.
I think 2 things will happen. The first thing is your sliced pan bread with a shelf life of 3 weeks is not going to go away. My expectation would be, and I'm partially extrapolating from what has happened in Europe, that you will see the continued use of ingredients and additives to keep the shelf life. Some of them will be replaced by a sourdough fermentation, which pertains to 10-20% of your wheat flour, which is then mixed up in a yeast level product. We may or may not see an increased production of locally produced bread, either supermarket or artisanal bakeries, and then we have the stuff that doesn't cost $2 per loaf, but it costs $4 per loaf. It doesn't have a shelf life of 3 weeks, but of probably no more than a week. And that's a question: are consumers willing to pay more for a higher quality product? And that's a question for the US I can't answer. For Canada and Edmonton, to some extent, I would say there seems to be a good chunk of consumers that are willing to pay extra for good quality. That's not everyone, but there is a substantial proportion.
Dr. Annor, just asking if you had any comments based on what Dr. Gonzale said and any of your thoughts.
Oh, okay. Yeah. I'm very happy to listen to the presentation from Dr. Gonzale. I think it's really very nice, and it actually kind of justify why we're doing what we're doing here in Minnesota with the whole thing of sourdough bread and how it reduces what you call ATIs and FODMAPs, and we are actually looking at it from the breeding standpoint. I think that makes it unique and adds on to what he has done so far. I think after this meeting, I would have to connect with you much more to make sure we are on the same page and not do things that are the same here in the US and Canada.
Brian?
More than anything, I'd just like to thank Dr. Gänzle for writing letters of support for our research over the years and being such a resource for what's happening in the United States. He's really the go-to guy, in our opinion, and we've learned a lot from his work.
Well, I'm more than happy to present. It's from my perspective, really for the better part of me working in Canada until after 2005, nobody, farmers or industry in North America, cared for sourdoughs. That is changing, and obviously I'm glad about that, and I'm happy to support that. So thank you.
I guess Grandma did it right at the beginning. Scott, do you have any questions as chair of the council?
The high fiber that we talked about with the low FODMAPs and ATIs, what is that? Traditional wheat breeding? Is that gene editing? Is that?
I think Jim.
So what the source that we're using is from induced mutations. So there's no regulation surrounding that. There are a couple of companies that hold patents on that though, so we would have to get a license from them in order to market a commercial product coming from such wheats. So it's so non-GMO methodology can be used.
I have a question for both of the food science professors. In light of sourdough not having the shelf life that other breads do, is there anything that can be done that would be considered acceptable by the consumer to increase the shelf life?
I hope in 2 or 3 years I can give you an answer because we're working on that problem. We are working on the problem together with an industry partner, which is a large North American bakery. I would think that a combination of sourdough, either with plant extracts with antifungal activity, and hops have been used in the past, or in combination with fermentation processes which get clean label propionic acid. And now you're cheating a little bit, but it's a fermentate and not an additive. A combination of sourdough plus something which maybe can be brought in alignment with a clean label and consumer acceptance will get you the storage life that industry needs. And there is a second aspect which is completely unrelated to fermentation. The fungal spores that spoil the bread always contaminate after baking, which means all you need to do to prevent fungal spoilage is make sure that anything between the baking oven and the packaging is done in a clean bench, avoiding each and every fungal spore to get inside your product. That problem is not easy to solve, but at least for one bakery in Europe, it has been solved at a large scale. I'm not sure about the economics of that, but probably, it's a combination of sourdough plus other antifungal ingredients, natural if it needs to be, plus improved hygiene that may get you to a 3-week storage life, shelf life without having a long list of additives and a strong smell of propionic acid.
So it's actually, he said most of the things I was going to say. I was going to come from the aspect of packaging. Like, is there anything that can be done with the packaging? Even if you have the fungal exposure to the bread exposure to the fungal spores, can you use some modified atmospheric packaging conditions to slow down the growth? That is something that could be considered. When you talk about packaging, it makes it a little bit expensive. So as Dr. Gonzale said, you end up with a product that may be out of the reach of people who may normally buy and consume them, but your packaging could be something that could be done. That could be modified or used to extend shelf life.
Becky, you got a question?
So when you look at different types of cultures, like a type 1, type 2, type 3, do they offer different types of benefits? Is one better than the other, or can you elaborate on that?
We don't know yet. So simple. Because we have enough data. We have enough data by now to have some confidence that sourdough modifies ATI, and that seems to be pH dependent. We have enough data to say that sourdough reduces FODMAPs, but to make a systematic comparison on which organisms you need, which fermentation times, and which proportion of the wheat flour needs to be fermented to have a substantial impact on the FODMAPs in bread, I would think it's too early for me to say anything definite about that. I do know that different sourdoughs are different, but I was surprised on Monday. I was reading the paper from Holland that I briefly referenced as a DOI on the slides, and they had a yeast fermentation with a 2.5-hour proofing time, which is probably at least 2 times as long as what's used by industry. But that apparently was enough to get a fairly substantial reduction of FODMAPs in the bread, and I wouldn't have predicted that. So we learn as we go.
Yeah, and I think to add to that, I think the main difference between the type 1 and the type 2, or the traditional and the one used in industry, is more of like the industrial one is much more purified. So you are likely to have a much faster rate of fermentation, and therefore you'll be able to slow the fermentation time. But yeah, as to the effect, is basically based on the fermentation time, is based on your initial microbial load for the fermentation. So that, as far as I'm concerned, that is the differences. It has to do for the industrial type, they want to cut down on fermentation time, of course, to improve output. So that's the main difference.
I want to thank everyone for participating today. Dr. Anderson, Dr. Annor, Dr. Gänzle, appreciate it very much. Brian, Three Farm Daughter Company, and folks there, thank you. And again, thanks to the Department of Agriculture, the Minnesota Wheat Research and Promotion Council for their assistance on this project. And George and Jim, we're looking forward to phase 2. We have a little bit of work hill to climb before we know for sure whether we can move forward with that research, but I sure hope we can, because there's a lot of potential here that needs to be uncovered. With that, Becky, is there anything I missed?
No, Harold, you did a fine job. And I'd like to thank the team and the researchers for all the work that they do to drive opportunities for the wheat industry. So thank you for that.